I've got the models. So the models I have up here are found on this PowerPoint, and I'll go through them. So I've taken pictures of them, and right, it's basically different phases of mitosis, right? And it even starts with interphase. The first one has interphase, and the last one is interphase 2. And I'll explain how, but these four in the middle are prophase, metaphase, anaphase, and telophase. Okay. Now, we talked about this in class, right? The cell cycle is a combination of two parts to it, right? Interphase and mitotic phase. The interphase has 3 parts, right? There's the G1, S, and G2 phases, right? And each one of them lasts for approximately 6 to 7 hours. Then when you get done with those phases, then you enter the mitotic phase. The mitotic phase is about 2 to 3 hours, something like that. It's probably different for different cells. And then there's 2 main parts to the mitotic phase, mitosis, and then at the end you have cytokinesis, right? But they belong under the mitotic. Then mitosis is divided into phases, right? Prophase, metaphase, anaphase, and telophase. Okay, so all of them. It's nice to know how this works for, when you're taking test questions. Then we're going to use 2 different, well, there's actually 3 altogether. When you use the term chromatid, then it's referring to chromosomes that are going through either mitosis or meiosis. And specifically, you say they're sister chromatids because they're in pairs. If you see 2 here, that means that this chromosome has been replicated to a sister chromatid. That occurs during the S phase of interphase, that's when they are replicated or duplicated. When you're looking at something like this, then we're viewing them during metaphase or during mitosis. It's gotten that far. So chromatid is describing them during mitosis. So is chromosome. If I asked you about the DNA on the 4 in the middle, right, they would be referring to, right, there's DNA, they look like worms, gray worms. And every model in the middle, right, you see those. Now they're gray here, and then this is in metaphase. But you would call them chromosome during these 4 phases, and then these 2 on the end represent, these cells now have gotten to interphase. This cell is still in interphase and it hasn't got into mitosis, right? So if I asked you about the DNA on these models, the correct answer would be chromatin. It is just a minor point. So a karyotype is a display of chromosomes during metaphase. So that means they've arrested cells that are in metaphase, and it would look different than what you see in this model. But they're spread out, and they'd be all over the photograph. And what they do is they actually cut them out of the paper of the photo and then put them together. And then when they made the karyotype, they said the largest chromosome was number 1, smallest chromosome 22. And that's how when you see this, it's a display of the chromosomes. So this first picture is of this guy, right? And if you look, the 2 little dots, the black dots, are centrosomes. So to get to this, there's normally just 1 centrosome in the cell, right? They are duplicated, so you have 2. And they're still stuck together. the next picture, you see the two black dots have separated, right? Then now we're into the next phase, which is a prophase. But that's how you would distinguish this one from this model, is that where the dots are. They look fairly similar other than that.
25 is our asters.
Yeah, well, no. 25 are the spindle fibers. Microtubules or spindle fibers. Now it's the centrosome here, it's still considered a centrosome here, although it's turning into the aster. These guys, after this step, I don't know, I might still label it a centrosome. If you called it a centrosome, I wouldn't mark it wrong on the practical. But, right, they turn into asters. And they're making these spindle fibers or microtubules, right, and they'll eventually connect to the chromosomes. So this is an animal cell. These are animal cell models. The metaphase is the easiest one to see. Yeah.
Why was one a chromatin and the other one was a chromosome?
Just the way it is. No reason. So here it's chromosome. You just need to, if I asked you on the test, what do you call that? If you called it chromatin, it would be wrong. Right? If I asked you, right, what's the structure here? If you put chromosome, it would be wrong too. You have to put chromatin for this case, chromosome when you get into the four. Right, that's what this said, right? Chromosome versus chromatin. But you do need to pay attention to those details. It's just part of science. So now I've got an arrow to the spindle fibers. They don't really show it all that well being attached to the chromosomes, but some of them are attached to the chromosomes and some of them aren't. Right? This is metaphase, and then this is anaphase. Now, the sister chromatids have separated. The sister chromatids have lined up in metaphase during mitosis, and then the sister chromatids separate during anaphase. Right, now, see, I don't label, this is technically, these two are asters at this point. Well, I actually put an arrow here. This one's an aster as well. Right, they're chromosomes, but pretty straightforward. Now, telophase, right, now the chromatin or the chromosomes, they've reached, they've gone past, and you get the cleavage furrow. That's the really identification of saying you're now in telophase. You infer the DNA structures as chromatin again, and you need to recognize the difference between this model and that model, right? People confuse this. This is interphase, not telophase. But you still have the spindle fibers now. That's what the lines are. You don't see any spindle fibers in this. And technically, probably at this point, you'd stop calling them asters. You'd go back to calling them centrosomes. I'm not sure there's a question about that, but that would be the correct. Now, this is another type of animal cell, and this is a picture from a microscope, and they've stained the chromosomes in the middle so that they look purple. Then this is a huge aster here. This is another huge aster. So it depends on the animal, what you would actually see. This is, I think, whitefish blastula. So at this point, they're very big. The chromosomes look pretty small in comparison. And I would guess that there's probably about 15 chromosomes in there lined up, or 16. I guess it'd be an even number. But that'd be metaphase mitosis. And then here's a few other, that's anaphase. It looks sort of like that, right? These are the chromosomes, and they have that kind of look to them. Metaphase, the chromosomes are still stuck together. And then here's telophase. They've separated, and you can kind of see small cleavage furrows there. It's not real clear on this picture. And when you look at these under the microscope, it's not going to be entirely clear. But if you're not sure and you want some help identifying what cell's what, let me know. This is obviously telophase it's probably just about done with cytokinesis. Right? Definitely anaphase, metaphase, anaphase in the animal cell. So you might see on the practical this picture and then an arrow pointing to that, and you might be asked, what phase of mitosis is that? Or this one, right? This one you can kind of see that they're still stuck together in the middle, but it's not that clear. But that's definitely anaphase, right? If you started to see a cell wall, like see here, now the cell wall is probably complete. This one, I don't see a good telophase. There, I think on this one you see a good telophase because, see, that's the cell wall there, or it's the cell plate, it's called, which in turn becomes the cell wall. But you refer to it as the cell plate during telophase, right? Obviously anaphase. And then these are plant cells, right? Which is what you'd expect to see, right? On the onion root tip, it's a plant root, and the cells are mitosing only in a certain part of the root, right? These are finished mitosing, these are mitosing, and as this thing grows in length, right, it just stays so close to the tip, the cells that are mitosing.
So is this picture showing us that there's multiple stages going, or phases going on?
Yeah, each cell, you know, like this is probably in prophase. Once you get past, you know, these cells are no longer mitosing in them. So it's only certain cells that are being told to divide. Right? I don't know which way the tip is on here. If this is an onion root, normally they're roots, because that's where you get a lot of mitosis in the plant. Right? As long as the root is growing, right, it's trying to get longer. So you'd have to find a plant that was occurring too. Once the root stops growing, then mitosis would stop. The purpose of the mitosis in the plant at this point is to grow the root. So it's a complicated concept, right? It's not, it's so simple.